my name is Aisha.
Hi, my name is Aida.
Hi, my name is Evelyn.
Hi, my name is Alice. In conclusion, the experiment helped us to understand the principle of spectrophotometry, highlighting the calibration curve that had been made to determine the concentration of BSA protein standard solution. We also have applied the analysis technique for qualitative and quantitative. Spectrophotometry had enabled scientists to examine a variety of samples without coming into contact with their skin because the samples are enclosed in a small tube known as cuvette. Both qualitative and quantitative analysis are possible with spectrophotometry. Light absorption increases as a substance concentration increases, while light transmission decreases. The experiment was divided into two parts. For the first part, it deals with the use of spectrophotometer cuvette to determine the absorbance spectrum for each test tube. The data that obtained from the experiment were used to calculate the average and standard deviation for each point, and then the graph of concentration versus absorbance was plotted. For the second part, the data for protein concentration determination were given in the table. A standard curve for the protein assay was plotted and the line of the best fit was perceived. The concentration of the unknown protein sample can be estimated by using the standard curve that was plotted. As a result, the objectives of the experiment were achieved. From conducting the experiment, it have helped us to understand the principle of light absorption and the difference between transmittant and absorbent. Other than that, this experiment allows us to recognize that many substances with low natural absorbance can react quantitatively with specific reagents, for example, the Bradford reagent, to produce a measurable product. We find it difficult to maintain the integrity of the measurement because of some factors that can affect the readings. For example, using a scratch or contaminated cuvette and avoiding bubbles when micropipetting for a more precise volumes of BSA protein standard. To improve our overall experiment, it is advisable that people should have a session for practice pipetting specific volume using various micropipettes before starting the practical for the standard curve. Furthermore, the solution of the unknown sample can be prepared by more than 2 to provide a more solid foundation for calculating the average and the standard deviation of the graph.